Hey everyone, it's Dr. P. We're gonna tackle the membrane structure and function chapter. It's chapter 7 in Campbell Biology, edition 10, but use whatever book you've got. So really what we're going to talk about is the borders of cells. So, and specifically the cell membrane, not the cell wall in plants, but specifically the cell membrane. And then what we're going to talk about is what's really important to that membrane, and that's how it regulates what comes in and goes out of the cell. So we'll look at things that can just cross through the phospholipid bilayer, but we're also going to look at some proteins and how they facilitate who gets in and goes out of the cell. First big concept you need to grasp is selective permeability. And that is an attribute or a property that is attributed to the plasma membrane. Selective permeability is just kind of how it sounds. So selective, it differentiates, it determines who can and who can't. And then permeability is crossing that membrane, who's permeable, who can pass through the membrane. So selective permeability allows some substances to cross and some to not cross so easily. It helps to maintain, and this is really the key part, why as to why it exists, but it helps to maintain different concentrations on opposite sides of that membrane. So the concentration of something inside the cell versus the concentration of that same item outside the cell. And that becomes really important for us when we talk about diffusion, when we talk about osmosis, when we talk about any of the passive transports, but even certainly the active transports and spending ATP to move stuff. So we'll talk about that later this chapter. New vocab term, I need you to get actually two of them off of here. Amphipathic. So amphipathic means that a molecule has both a hydrophobic and hydrophilic portion. So, and you know this already because you know that the plasma membrane is made up of a phospholipid bilayer. And in chapter 5, we talked about phospholipids and that we had that polar head and those non-polar tails. So the polar head was hydrophilic and the non-polar fatty acid tails were hydrophobic. Okay, so that's amphipathic. Your second term that you need to pick up here is fluid mosaic model. And fluid mosaic model is, it talks about various proteins and it even talks about the phospholipids themselves within that plasma membrane. So it talks about the fact that the membrane structure is fluid, so that items within the membrane are movable, and we're going to look at that in a second. And the mosaic portion is that it's not just one thing. So if you think of a mosaic made of tiles, or you think of another term for mosaic, or even those photos, those large giant images that are made up of teeny tiny other photos, that's a mosaic. So this phospholipid bilayer isn't just the only component to the plasma membrane. It has proteins and glycoproteins and other stuff embedded in it. And we'll look at that in just a second. But before we do, I want to remind you of what that phospholipid looks like. Okay? We've chemically analyzed them. We know that membranes are made up of proteins and lipids. Sometimes they're bound together in a phospholipid, in a glycolipid, so that we've got a glucose or something attached to the lipid, or even a protein embedded in there, and that looks something like this. So you have that phospholipid bilayer, and then you're going to have proteins embedded in, okay? So they'll sometimes cross the whole width of the plasma membrane the way these appear to, or sometimes they only go partially across. So they can be, and we'll talk about those different terms. But we know that fluid mosaicism happens, that this model is accurate, because we did some freeze fracturing, which is freeze fracture studies, which basically we do a specialized technique. We freeze that phospholipid bilayer and then we split it in half. And it literally splits between those tails right along the middle, right through the middle of the peanut butter, if you will. So if this is bread out here and this is bread out here and the peanut butter's in the middle, it splits right along that middle part right there. And the proteins ended up going on one side or the other. Now, phospholipids can move within that bilayer. So this phospholipid, we'll call him 296. 296 is not stuck here rigidly between 295 and 297. 296 can move over here, he can migrate over here, he can end up on this edge, he can end up all the way over here. So these phospholipids are migratable, and these proteins can shift as well. So the component pieces can move as well. And they typically will drift laterally, side to side, like this way. They don't typically flip. So 296 here does not usually come over here and hang out with these guys. It can happen, but it's rare. It's not common. They're on one side or the other, and they don't typically flip-flop across that membrane. Some keys, remember that you're dealing with a lipid. So you're dealing with a glycerol and those fatty acids that we talked about, right? So as temperature cools, those membranes move from a more fluid state to a more solid state. Okay, how fluid or how solid really does depend upon temperature. And if we're cold enough, we can actually freeze or get those guys solidified. And that depends upon the temperature, but also the type of lipid that's present. So if you're rich in unsaturated fatty acids, then you're going to be more fluid than those that are more saturated. And we talked about that when we talked about saturated and unsaturated fatty acids. They have, those unsaturated have the bent tails. And if you can see that, those bent tails are going to prevent, you can't see me moving my hands, but I really am for you guys. So they prevent those phospholipids from being jam-packed super tight together. They've got some space, they've got some room to move, and that makes them more fluid. Whereas if you had all saturated hydrocarbon tails, then they're going to pack really tight, like stacking pencils in a box. You can lay them out and you can get them really tight, and once you cram them in there super tight, they don't move so well. So that fluidity of the membrane is in part due to the composition. How many saturated and how many unsaturated fatty acids do I have? The other thing that really, really lends itself to maintaining this membrane, because remember, it's typically, and maybe I didn't say it, but it's typically about as fluid as salad oil, okay? One of the things that helps with that is a steroid called cholesterol. So you may or may not have known that cholesterol is a steroid, but the steroid cholesterol has some really different effects on the fluidity of the membrane depending upon the temperatures. So in warm temperatures, 37 degrees Celsius, for instance, the cholesterol retains or restricts, I guess is a better word, the movement of those phospholipids. It keeps them pretty much where they need to be, okay? It doesn't let them run like salad oil. Your cell membranes are not running around like heated salad oil or olive oil. At cooler temperatures, what that cholesterol does is it's a space taker-upper, and it prevents those phospholipids from solidifying and packing in super tight together and making those cell membranes too rigid. So it looks kind of like this. So this is an animal cell. And what we've got is we've got some bent tails here and there, and we've got these cholesterol embedded in between those phospholipids. And again, what I really want you to know is I want you to know, what does cholesterol do? So what does cholesterol do at warmer temperatures, and what does cholesterol do for a membrane at cooler temperatures? Okay, so there you go. Just like I said, what structural features are found in a plasma membrane that makes them more fluid? And then how does cholesterol affect membrane fluidity at, number one, warmer temperatures, and number two, lower temperatures? So pause your video, answer your questions. I'm going to give it a count of 5, and then I'm going to flip to the answers. Okay. There are your answers. If those don't make sense or you're struggling with them, then either hit me up in class or shoot me an email, Canvas message, whatever, and we'll figure it out. Okay, so membrane proteins. So I've been talking I started this chapter saying, oh, we're going to talk about the proteins. And then we just spent all that time, the first 10 minutes, 11 minutes, whatever it was, talking about phospholipids and barely even touching proteins. So now we need to grab those proteins. So the membrane has all these different proteins. Remember, it's mosaic, and they're embedded right into that fluid matrix of the lipid bilayer. So they're embedded right in. And these guys are going to determine a lot of the very specific functions that that membrane is or is not capable of doing, just depending upon which are present and which are not. So this is from the textbook. I cannot take credit for this in any way. This is much too pretty to be my work. But here is the phospholipid bilayer. In it, you can see the cholesterol that we talked about and what those did. Here are some proteins that are embedded. Here is a glycoprotein, meaning I have a carbohydrate attached to one of those proteins. Here's a glycolipid over here. Sorry, here's a glycolipid over here where I have a glucose or a carbohydrate attached directly to one of the phospholipid heads. Okay, and to orient you, this is inside the cell. So down here where it's kind of sandy, beigeish, orangish, this is inside the cell. This is outside the cell. This is called the extracellular matrix. And it's made up of a bunch of stuff that we'll talk about. But right next door, and this looks big, right? This looks huge. It looks like there's a great big gap before, and you don't even see another cell. But in reality, this would be a microscopic space, and you'd have another cell membrane right there on the opposite side. So don't think of it like the distance between planets, because it's not. So let's talk about those proteins. We have two types of proteins, or we name them based on their location. One are peripheral proteins and the other are integral proteins. Peripheral proteins are bound to the surface of the membrane. They don't cross the entire distance. This is not peripheral. This is. See these guys? They're bound to the surface. So these are peripheral membranes. And looking at it from the outside, you might not be able to tell that. But these are peripheral proteins. They're bound on either one side extracellularly or intracellularly. The other are the integral proteins that we talked about, and that's these guys. These guys are integral. They're integrated into the membrane, and they cross the entire phospholipid bilayer. So that's the important difference between them. The hydrophobic portions of integral membranes are lined up with the fatty acid centers, and the hydrophilic portion are going to be lined up with the polar heads. So this, and I'll show you in a couple of slides here, you're going to have multiple regions within this protein. You're going to have hydrophobic and you're going to have both hydrophilic. And that's important because it's going to be key to how this membrane functions. This is my little visual reminder to just remind you guys to be thinking about proteins. And remember, they're not just a blob. They're just not like, I don't know, a plop of whatever on your plate. That isn't how they work. Remember, they have structure. Remember what we talked about with primary and secondary and tertiary and quaternary, and that we can have multiple strands that are pulled together to make a very complex protein. And this is just an artist's rendition of a protein, an integral protein embedded into the plasma membrane. All right. 6 major functions of membrane proteins, and now these aren't just proteins, these are membrane proteins. So transport, they're going to help us get stuff out of the cell. Enzymatic activity, they'll actually function as enzymes. Will be a signal transduction, so that you're going to trigger a signal from something extracellular to generate a response somewhere intracellularly. Sometimes there's cell-to-cell recognition, and that's how cells know to bind to one another. Remember the visual that you had with the antibody and the influenza B, okay? And how that antibody could only bind to that influenza B. So you get that cell-to-cell recognition. We're actually going to have proteins that are going to join cells together. And then lastly, we'll have that attachment to the cytoskeleton. So they're anchoring points, both inside and outside of that cell membrane. All right, I'm going to take a little break here. We're going to stop this video, and I'll pick up on the next one. So I'll back up one slide, we'll reintroduce that, and we'll go from there.